Bonjour tout le monde. Mesdames et messieurs, je me présente, David Eidelman. I'm the Dean and Vice Principal Health Affairs, Dean of Medicine, and it's my great privilege to welcome you to the 9th installment of the Holmes Lecture. So for those who don't know, Holmes was one of the founders of the Faculty of Medicine and the first official Dean of the Faculty of Medicine. And we use the Holmes Lecture as an honor lecture to invite people to present really cutting-edge stuff. And we try and pick speakers who have something to offer particularly for our students, our fellows, and even for faculty members. I think in today's case, we have a speaker, Dr. Roden, who I'll go into detail in a second, who actually is very consonant with Dr. Holmes, because some of you know that although in those days there were no cardiologists, Dr. Holmes was actually interested in that, and there's a famous congenital malformation called the Holmes heart, for which he is known, and apparently we have that in a jar somewhere. So more seriously, I really want to acknowledge the Rose Weiselberg Foundation, which actually is providing the resources to make this lecture possible. That lectureship was established in the honor of Rose Weiselberg, who was actually, as a pulmonary doctor, she was one of the first women pulmonary doctors in Montreal, and the foundation was established by her sisters in her honor many years ago. And the goal is to salute those who are outstanding in the health sciences and who can help us celebrate what's really the backbone of our activities in medicine here, education, research, and healthcare. I'm glad to see lots of students in the audience, particularly on such a rainy day, because it's very important that our lectures be open and accessible to our students and other and postdocs. So in that regard, I'm really honored to be able to present to you Dr. Dan Roden. He's professor of medicine, pharmacology, and biomedical informatics, and director of the Oates Institute for Experimental Therapeutics. He's also a senior vice president for personalized medicine at Vanderbilt University. So like me and like many in the room, he's a native Montrealer. He received both his bachelor's and his MD-CM degrees from McGill and did his internal medicine at the Royal Victoria Hospital when it was located across the street. He went on to do clinical pharmacology and cardiology at Vanderbilt as fellowships. And once he was there, he stayed there. And he's been at Vanderbilt ever since. And his career is focused on the study of clinical, genetic, cellular, and molecular basis of arrhythmia susceptibility. And so this is an area that's both of great clinical import, but also great scientific interest. And in particular, the variability of responses to antiarrhythmic therapies. In 2006, he was named to lead Vanderbilt's efforts in personalized medicine. And over the past 10 years, he's led a broader effort at Vanderbilt in pharmacogenomic discovery and implementation. In particular, this has included directing the Vanderbilt DNA data bank, which is called BioView, or BioView, I guess would be how you pronounce it. I'm sorry, you're in Montreal here. Et on parle français, c'est là. A discovery resource that as of early 2017 included more than 230,000 samples linked to de-identified electronic medical records. He's also a leader in Vanderbilt's PREDICT project, which since 2010 has been preemptively embedding pharmacogenomic invariant data in electronic medical records of over 14,000 Vanderbilt patients. He's the recipient, as you can imagine, of many honors and awards. Those include the Leon Goldberg Young Investigator Award, the Rawls Palmer Progress in Medicine Award from the American Society for Clinical Pharmacology and Therapeutics, and the Distinguished Scientist Award and Douglas Zipes Lectureship from the Heart Rhythm Society. He's also a recipient of the Distinguished Scientist Award and the inaugural Functional Genomics and Translational Biology Medal of Honor from the American Heart Association. He currently serves on the Advisory Council to the National Human Genome Research Institute and the Science Board of the FDA, and has been elected into the membership of the American Society for Clinical Investigation, the Association of American Physicians, as well as being a fellow in the American Association for the Advancement of Science. So truly an extraordinary number of honors for an exceptional person, and we're thrilled and privileged to have him come and speak to us today. Without further ado, Dr. Roden.
Thank you. So I'm thrilled to be back here. I've told somebody today that this was the first time I've been back to McGill to talk, but seeing Dr. Fuchs, I'm reminded that at my 25th anniversary reunion, I did actually give a talk up in the Round building. So this is the second time I've been back. The audiovisual guys tell me that I have to keep on looking at them. There's this overwhelming temptation to look at the screen, and I'm not allowed to look back there. So this is awkward for me, and if I feel sort of tongue-tied or eye-tied, you'll understand why. I have too many slides, and I didn't actually figure out whether the audience is students or physiologists or administrators. It seems to be a lot of both, a lot of most. So bear with me if you find it simplistic, and bear with me if you find it too complicated. So this is the title I've given myself. I actually, when I talk in the United States, it's just much easier to use the word, the degree MD. But for here especially, I like to use the MDCM. That's a McGill degree, and I'll have more to say about me and McGill right at the end. So if you take a population, I'm very fond of this graphic because obviously when I'm in the States, it's a different map when I'm in the States and Canada, but it's sort of, I make a hobby of making different maps. So if you take a population and ask the question, what happens when you give a drug to a population? Some people have a good response. We hope a lot of them, but some people have a good response. Some people have some side effects. Sometimes nuisance, sometimes more than that. A couple of people will have catastrophic side effects. And then there's a whole bunch of people still in white because nothing happens. I'm fond of saying the most serious adverse event, or the most common adverse drug event, is that there's no response to the drug. That's the most serious event. So that's what happens in a population. But you're the patient who's sitting in the doctor's office. You want to know which of those people you are. You don't want to know what the population is. And so we've understood over the last decades of work that there are lots of things that influence how you respond, whether you're old or young, your ancestry, your food intake, your environment, the other drugs that you take, genetics. And those are the things that we've focused on over the last couple of decades to try to sort out, and I'll try to sort of give you a sense of how that works. So this is big data and little data. I arrive at Vanderbilt about 6 weeks after this paper is published in the New England Journal of Medicine. The first author was a fellow when the work was done and was a newly minted assistant professor, so way senior to me. And he was my mentor, and of course over the years we became good friends and colleagues. So what Ray Woosley did was when he was moonlighting, he would look at he would look at paper records and of patients who were exposed to a drug called procainamide. Now, a common side effect of procainamide is that people get the drug-induced lupus syndrome, arthritis, and a number of other manifestations. And he tried to sort out whether there was an influence of genetics on the development of antinuclear antibodies, which is the biochemical hallmark of the lupus syndrome. And he did. How he acetylated people, how he genotyped people is complicated, I won't get into. But this is the graph that appeared in the New England Journal. And the important point is that 21 patients made a New England Journal of Medicine paper back then. That's small data, and this finding sort of holds up, that in fact if you're a rapid acetylator, you're less likely to get lupus, and that's from Ray's original work. My first project was to work on a drug called encainide, and when we looked at encainide, we looked at lots of different things, but one of the things was pharmacokinetics. We gave everybody a 25 milligram dose of encainide at the beginning of the study and followed plasma concentrations, and then tried to relate that to drug response. And what we had was 10 people who had a really gratifying drug response. I won't get into the details because it's pretty fuzzy over the next couple of decades. But 10 people had a demonstrable pharmacologic response. And what you can see is they're in blue up there. And again, I'm not allowed to look, right? They're in blue, and I'm supposed to use the pointer, but I'm not going to do that. They're in blue. You can see it. And their half-lives are all pretty rapid. They all eliminate the drug very rapidly, but the peak concentrations vary from like 1 to 100 nanograms per ml. That's an enormous variation in the peak concentrations. And there's one non-responder. And the non-responder had the highest concentrations we saw, the slowest drug elimination, and we were really at a loss to explain that. I worked with some of the most brilliant pharmacokineticists of our time, and they had lots of complicated explanations which were completely wrong. The explanation is that encainide is a prodrug. and it requires bioactivation, requires metabolism in the liver to something that is active, and it's the something that is active that produces the pharmacologic effect. If you're unable to metabolize encainide, you achieve very high plasma concentrations of the parent drug, very slow drug elimination, and no pharmacologic effect. So, and the name of the enzyme is CYP2D6, and it's one of the poster children for modern clinical pharmacogenetics. And again, little data. This was also in the New England Journal of Medicine. 11 patients gets you into the New England Journal of Medicine back in 1980. And this is the third example that I want to give you. It's work that actually was all done by a couple in Australia, but now they're at Vanderbilt. I'm going to claim credit for them. So this is the Stevens-Johnson syndrome, a really horrible drug reaction. Can be fatal. It involves the skin and mucous membranes, and these are pictures meant to disgust you. And what Simon Malal did in the early 2000s was that he figured out that there was a very, very strong association between a particular HLA haplotype and the development of the Stevens-Johnson syndrome on exposure to an antiretroviral drug called abacavir. And you can see the odds ratios there. When the odds ratio creeps above 5 or 10, we get excited in genome land. And so when the odds ratio is like 822, you get very, very excited. So that was not enough to convince the HIV guys to start to test people genetically. So he and his scientific colleague and now his wife, Elizabeth Phillips, mounted a very large trial where they basically exposed 2,000 patients who were starting antiretroviral therapy to abacavir. They randomized them to conventional therapy, so that would include abacavir, or genetically guided therapy, which would include abacavir unless they had the HLA risk allele. And then they counted skin rashes. And in the conventional group, there was, I have to look at the numbers exactly, it's about 8% people got skin rash, and in the abacavir group, there were about 3% that got skin rash. But what Elizabeth had done was develop a way of actually looking at the rashes biochemically and immunologically and figuring whether they were abacavir related. And when they did the skin test to demonstrate that, there were absolutely no cases of abacavir-related skin rash in the genetically guided therapy group, and about 3% in the conventional group. So that's become standard of therapy. So it's not only a predictable adverse drug reaction, it's a preventable adverse drug reaction. There's lots of subtleties around this which I don't get into. So the big paradox in personalizing medicine, if you want to use this genetic or other kind of personal information to actually drive care that is not average for an average person, but actually somehow personalized to what that person is, you have to have a very large denominator to draw from. So you start with 100 people, and you have one who has some aberrant genetics or aberrant physiology. It'll be very hard to convince anybody that that person should be treated differently. But if you start with 100,000 people and you have 1,000 people in that funny little subset, then you can start to develop the evidence. So the paradox is that in order to develop personalized therapy for that one person, you have to start with 100,000 to develop the subsets. That's the idea. So our vision is encapsulated by this cartoon. Every so often I say to myself, I should stop showing this cartoon, but it really resonates with me more and more that this cartoon came out in the New Yorker in 2000 when the first draft of the human genome was announced in the White House. And the woman is handing her sequence to a pharmacist. So she's not handing it to a doctor, not handing it to a nurse, a pharmacist, so it's a healthcare team. She's handing it to him on paper, which is not the way it's going to happen, I'm pretty sure. And he has no idea what to do with it, which is about where we are right now still. When Francis Collins became director of the National Institutes of Health, he was interviewed by the New England Journal and had lots to say about lots of things, including this. This is the sum total of what he had to say about pharmacogenetics. And what he said, basically, you read in the red box, it says, if everyone's DNA sequence is already in their medical record, it's simply a click of the mouse to find out all the information you need. Now read down to the bottom. It's not going to be terribly expensive. Yeah. And it should improve outcomes and reduce adverse events. So we sort of buy that Kool-Aid, we drink that Kool-Aid, and we believe that, with one exception, and that's this word. I mean, it is absolutely not simple, but we're working on it. So this is a picture that I owe to my then young colleague, he was a fellow when he took this picture, Josh Denny, who's now my partner in crime in many of the things that I do, he had the foresight as a biomedical informaticist to take a picture of what the medical record room looked like before there was an electronic medical record. So I'm fond of showing this. I don't think it exists anymore at our place. It may exist at other places, and luckily it doesn't exist. So we started to develop an interest in investing in this whole area of personalizing medicine, which I will say most of what I'm going to talk about is genetic, but there's a whole piece of this that is not genetic, and I'll come back to that a couple of times during the talk. So if you search for Vanderbilt and personalized or precision medicine, this is the number of PubMed hits. And I'm very proud of the fact that the first one was in 2008, and the first one actually described the architecture of our biobank. And each one of these people who were authors made huge contributions to how we designed and deployed that thing. And so fast forward 8 years after that publication, last summer we were named by the NIH to run the data and research hub for the Precision Medicine Initiative. That was a $75 million grant, largest by far grant in Vanderbilt history. And I think it's sort of, the biobank was one of the things that propelled us into this whole field, into understanding how to do this. And the development of the informatics expertise around the biobank, I think, is one of the things that made us into this hub. I'm fond of saying, it says down here, Vanderbilt Google to build Obama Precision Medicine Cohort. We have a couple of junior partners. One of them is the Broad Institute. One of them is Google. I'm happy to call them our junior partners, though. It makes me happy to do that. So BioView by the numbers, or BioView by the numbers. We start with the electronic record and we de-identify it. There's a whole science to that, which I'm not going to talk about, but it's about 2.5 million records. And we can search that and do interesting things with that. But a subset of those people have DNA samples. And every Monday I get an email telling me how many samples there are. So this is this Monday's number, 237,000 and change. And the seamy underbelly is that they've not all been genotyped or sequenced. But we're in the middle of a large project, which we call the mega project for technical reasons. The chip is called the mega chip. That's what we call it, the mega project. And we'll be over 100,000, hopefully by the end of this year. And that'll start to let us do things that they're only a glimmer in our eye right this instant. So one of the capabilities that we developed pretty early, which is lots of fun, and if I had the nerve, I would demonstrate it in real time, is something we call the record counter. So an investigator says to themselves, I want to know how many samples we have with X. Now, you can't do detailed searches. If the phenotype is complicated, you know, how many people got this drug, followed by this rash, followed by that drug, followed by some renal function test. We can't do that. But what we can do is we can search by codes, and we can search by a little bit of natural language processing. So here's an example. In the time it takes me to show the slides, this is how long the search takes, because the entire 2.5 million records are on a specialized search engine, so the searches really happen in real time. When we started this, they didn't. You'd send your search in, and 12 hours later you get an email saying, you know, go look at what the result is. But now it's all immediately in real time. So this is a search where I ask the question, how many people do we have who take Plavix or clopidogrel, a common antiplatelet drug, and have genetic information? So search for the word clopidogrel in the medication list, and there are 41,000. I thought that was not enough, really. It's a pretty commonly used drug. So then the sad part of this story is that I then searched for the trade name, Plavix, and it turns out there are 68,000 or 69,000 mentions of Plavix, and the intersection is 74,000. And there's a reason that these two numbers don't agree with each other, and that number is not exactly. In this example, they are exactly the same. Sometimes they're not exactly the same. Actually, here they're not exactly the same. There's a reason for that. Anyway, some people ask me about that afterwards. So then I click down on this button that says how many of them are in BioView, and about 19,000 are in BioView. And how many of them have genetic information? So we go to this, and we actually ask about one specific platform, which is the exome chip that we were using at the time. And there were 31,000 people with that, and the intersection is 4,400 people. So within seconds, literally seconds, an investigator will know that they have 4,500 people on this drug and have genetic information. Now the challenge is to figure out what to do with that, and that's more than just a quick web search. So But we've sort of developed those kinds of capabilities. And then you can click on another button and it'll tell you how many people are male, female, African American, how old they are. That's important. If you're writing an NIH grant, you have to put this stuff in, so it's all done for you automatically. So I can stop now and then or talk for another. No, I'm not going to stop now, but I can talk for another hour and a half about what we've discovered, but I'm going to run through that very quickly because I want to talk about the future tense a little bit. So we can find cases and controls doing this, using this and other kind of search algorithms, more complicated. We can export the cases and controls to other electronic medical record systems. We were part of the electronic medical records and genomics network since the beginning, and we work with other sites that have similar biobanks, although I will say they're not nearly as big. And this word phenome-wide, I'll come back to that in a second because that's one of the really exciting things that we're doing. We can look for common SNP associations that are already out there in the literature and replicate them. We've done a lot of pharmacogenetic stuff. These are some examples. And we can reuse existing genotype data to ask new questions. So this is one of the cardiac conduction system stories. I'm not going to talk very much about arrhythmias, but I'll show you that one as an example. And there's that word phenome-wide again. So let me explain to you what that means. So an ordinary genetic experiment, whether you're studying a family or a large population, starts with a phenotype, starts with something that's wrong with the patient. And then you get people who have the phenotype and people who don't have the phenotype, and you do some kind of genetic analysis, and you come up with a set of genotypes that are associated with the phenotype. That's a genotyping experiment. We realized after we had been accumulating genotypic data for a while, and after we had this dense electronic record, that you could turn that experiment on its head. So we started this experiment with a genetic variant. We could ask, here is a single nucleotide variant, a single nucleotide polymorphism, a SNP. If a person is wild type or variant at that particular site, does that predict any particular phenotype across the spectrum of phenotypes that are contained in an electronic medical record? So here is our first example, probably still our best example. So this is a SNP that is in a gene called IRF4. It's been associated with hair and eye color. And what we did was we asked the question, does having this SNP associate with anything else across the phenome? So the way these data are displayed, I realize that not all of you may be familiar with this, is that every dot is a statistical association. There are about 1,500 dots on this slide. Most of them are sort of down at the bottom of this thing and don't reach statistical significance. The y-axis is the negative logarithm of the p-value of that particular statistical test. So if the p-value is 10 to the minus 3, you'll see a number 3 here. So what we have is an association between that SNP and the diagnosis of actinic keratosis with a p-value of about 10 to the minus 28th. So that's pretty statistically significant. We also have lots of associations with basal cell carcinoma and other kinds of skin cancers. And then we have this really interesting one down here. Solar dermatitis. I'm fond of asking the audience, what do you think solar dermatitis is? I'm not going to bore you with asking. Solar dermatitis is the way sunburn is encoded in the medical record. So this is a very fancy way of saying that people with red hair get sunburn more often. We also get a signal with osteopenia, and that actually might be related to vitamin D metabolism in the skin. So we think that that might actually be real. Yep. So one of my other triumphs, and I'll probably show you all the triumphs because I've had lots of things that didn't work as well as these. I have a friend who studies mice, and he has been obsessed by the way the cardiac impulse conducts in the mouse. And he has isolated Purkinje cells, which are the conduction system cells, and made DNA libraries of genes that are expressed in the Purkinje cells. And one of the genes is ETV1. And he knocked ETV out of mice and had a really interesting phenotype, sent it to Nature Medicine and to JCI. And they both said, well, that's really interesting, but what does it have to do with human disease? And I gave a talk like this at NYU, and I said, he came up to me afterwards, he said, can you look in your bank and see if anything is associated with ETV1? So we looked at SNP associations between ETV1 variants and the range of diagnoses that we extract from the medical record. There are about 1,000, 1,500. And we have two top SNPs here. You can see them right there. And they are conduction system disease. that was pretty gratifying. And then we did the same experiment in Caucasians, and we came up with heart block. So that's really, and so the paper actually got published last year in JCI. We're very interested in using these kinds of approaches to understand drug actions. So one of the common drug targets in rheumatologic disease is TNF-alpha. You scan the TNF-alpha gene and discover multiple SNPs, and then ask the question, are there associations between those SNPs and any of the diseases that TNF-alpha antagonists are used for? And there are very strong associations between some of the diseases and less strong associations, but they're there nevertheless with others. So we think that this is a way of finding new indications for old drugs, to predict adverse drug reactions, and to perhaps identify new drug targets. So that's a huge area, again, a topic for an entire hour of talking. And then this is a really geeky, complicated example. I have this in here for you and for you. Okay, so this is what I've talked about so far. A single SNP pushed across the medical record. What are the associated phenotypes? But it turns out that you can do this experiment many ways once you have the curated phenome. You can ask, I gave somebody a drug when they were 1 year old. What are the phenotypes I see in the 5-year-olds? Somebody has a disease. Are there other associated diseases? Or how about just multiple genetic markers all taken at the same time? Wouldn't it be cool if we could actually measure gene expression in everybody? So if I took everybody in this room and I said, what is the level of expression of my favorite gene in your liver? I might be able to ask the question, with what phenotype does variability in expression of that gene associate? Now, I can't do liver biopsies on everybody in this room. Probably won't let me do that. And certainly brain biopsies are out of the question. So here, how do you get to individual estimates of gene expression? And I have a colleague, Nancy Cox, who joined us about 2 years ago, and this is her experiment. So basically, it starts with a data set called GTEx. These are patients, recently deceased patients, who have RNA sequence data, so gene expression data in multiple tissues and whole genome sequencing. So because she's a statistical geneticist, she can work magic, and she can generate multi-SNP predictors of gene expression. And I'm fond of saying when I describe this experiment, if you had told me that we were doing this 10 years ago when we started the biobank, I would have said that's crazy. But these are the kinds of things that people can do now. So then we deploy that, and now we can deploy it across tens of thousands of people. Whoever has SNP data, we can guess what their level of gene expression of a particular gene is and ask with what phenotype does that associate. So, I heard this whole story, and I must say I had to hear it many times before I could sort of write it down on slides and convince myself that this was going to actually be feasible or work, and then we had a big success. I think it's a big success. So Nancy's first big success with a gene called GRIC5, until then not very well defined function. So she developed the predictors for GRIC5 expression, deploy them across the electronic medical record, and when we get these data in raw form, it's an Excel spreadsheet. So this is what the Excel spreadsheet looks like, a condensed version. And what you can see is that there are a lot of eye diagnoses. And I've looked at lots and lots of these spreadsheets, and it's unusual to see an eye diagnosis at all, let alone this whole collection of eye diagnoses. Remember, this is all Nancy Cox, the statistical geneticist, sitting in front of her computer. She's not doing any wet lab or anything. And so she goes up 3 floors and talks to the zebrafish people, and there's this technique where you can very rapidly knock out a gene out of the zebrafish genome. They do that, and as she says, it's been 20 years since I looked down a microscope, but even I could tell that one eye is a lot bigger than the other eye in the knockout, or one eye is missing in the knockout. So this paper and it took about a year to figure out what the mechanism is. It turns out it's a vascular mechanism, and that paper has been submitted. So that's a big triumph. And whether it'll ever work again, we'll see. But it's really an interesting kind of result. Now, I said earlier, not everything is genetic. I have a young colleague who's doing what I do, but in the Children's Hospital, and she got interested in acute kidney injury in children. So she used the informatics engine that we have to identify individual predictors of acute kidney injury in children, put them together into a multivariable model. The multivariable model performs not badly. This is a graph. For those of you who have seen these kinds of graphs before, you say it performs not terribly, not brilliantly, but not terribly. For those of you who haven't, you just have to take my word for it that that's what this shows. And the really interesting part is that we can now take this model and every time a kid comes into the hospital, on the fly, calculate the risk score for acute kidney injury. There are two risk scores, one for the kids in the ICU and one for the kids on the floor. And I don't know if you can, yeah, you can see that. So those are risk scores over time. And when the risk score reaches some threshold, you fire decision support. And what we've discovered, or not discovered, what we decided to do, which I think is pretty clever, is we fire decision support for odd-numbered records, and we don't fire it for even-numbered records. And so we will see at the end of November this year, which is when our first data look is, whether there's a difference in pre-specified outcomes. So you can do a randomized trial of a new tool like this in the electronic record, using the electronic record as the trial tool. So this is a set of slides that I put in last night. It may serve to confuse, and it may serve to enlighten. So this has been the vision, but do you really want to get sequenced? And I'm not sure what the answer is, because people are going to get this sequence data anyway. But there is something called Moore's law that everybody is or probably is familiar with, and that says that the cost of computing halves every 10 months, or the computing power in your PC or in your telephone doubles every 18 months. So this is Moore's law in genome sequencing. The first genome was sequenced around 2000. And the cost was $3 billion, they say. And if they did another one right away, it would have been a couple of $100 million. So for a while, the costs dropped according to Moore's law. And then in 2006, new technology comes along, and they really, really drop. And what's really interesting is that over the past little while, they've started to drop again. And we're going to see cheap genomes. Whether we have the analytical capability of handling them, that's another story. But we have cheap genomes. So the problem that raises is that everybody in this room, if you compare yourself to a reference, you have somewhere between 20 and 30 million places in your genome that are different from the reference. And which of those are important for your health and which of those are irrelevant is the huge and looming problem in genome medicine. Genome medicine meaning the art or science of applying the genome to healthcare. So, and the letters that I hate to hear more than any other are VUS, the problem of the variant of uncertain significance. And here's how we got into this in a big way. Through the Electronic Medical Records Network and the Pharmacogenomics Network, we developed a collaboration which we call eMERGE PGX. This is a side project of eMERGE PGX, but basically we sequenced 2,000 people. And we sequenced them in many genes, but two of the genes that we sequenced them in were SCN5A and KCNH2. Those are two ion channel genes. Rare variants in those genes cause something called the congenital long QT syndrome, a disease where young apparently healthy people drop dead. So that's a phenotype you would like very much to avoid, and I spent a lot of my time thinking about that problem. But out of those 2,000 people, we had 128 variants in those genes. Some of those variants are common. So common that you don't worry about whether they're disease-causing or not, because they can't possibly be disease-causing. And some of them, many of them, most of them, were rare. And so we asked 3 labs, 2 commercial labs and a research lab, if you got this result in a patient, what would you say to the patient? And the answer was, one lab said 16 were pathogenic, one lab said 17, one lab said 24. The agreement was not much. And that's one problem in genome medicine. But we're the Electronic Medical Records Network. So we decided to go into the electronic records. There's not much there. Some of the people don't even have ECGs. But of the stuff that was there, there's very little to get excited about. So there are lots of people who have lots of variants. These are only the patients who have variants that some lab thought was pathogenic, not the 128. So that's, we worry about that. This doctor is saying our statistician will drop in and explain why you have nothing to worry about. And I'm fond of showing this. Alvin Schreier has seen this before. I showed this at a Gordon conference, and before I said, does anybody know what this is? Before I could even finish the sentence, okay, somebody knows what it is. Tell me. It's Bayes' theorem, right. So that Bayes' theorem says that if you take a bunch of middle-aged healthy people and do genetic testing, and find something, it's probably just a false positive. It also says that if you take a 12-year-old who has a QT interval that is really long and do genetic testing and it's negative, they have something even though your genetic testing didn't find it. So it's all about prior probabilities and how the test adds a little bit. And that's where we're going to have to be. So having said that you don't want to get sequenced, we're sequencing. So this is something called the PREDICT program. PREDICT stands for this, and it's our attempt to start to embed pharmacogenetic variant data, which I think is actionable in the electronic record. So our plan was find people who are going to get a drug. So people who get knee surgery, at least when we started this, were always put on warfarin. People who got a stent in their coronary arteries are always put on clopidogrel, Plavix. So find those people and then genotype them not for the variant that's important for that particular drug, but for that drug and a whole bunch of other drugs, and then store the genotypes, develop the clinical decision support, and figure out what to do with that, and then track what happens to outcomes. So we're planning this process, and the FDA does us a huge favor because they relabel clopidogrel, the antiplatelet drug, to say, just read the bottom line. The bottom line says, consider alternative treatment or treatment strategies in patients identified as CYP2C19 poor metabolizers. Clopidogrel is like encainide. It's a prodrug. It needs to be bioactivated. It's bioactivated by CYP2C19. And if you have loss of functional alleles in CYP2C19, you'll have less antiplatelet effect. And how, I have to show this, and that is, I can show this anywhere in this talk. People want genetics to be yes or no. Do I have this disease? Do I not have this disease? Am I going to have a bad drug effect? Am I not going to have a bad drug effect? And what a lot of modern genetics is going to do is going to shade the odds. And to the extent that the odds, we're shading the odds for some important outcome, the genetics will be important. To the extent that we're shading the odds for something not very exciting, it'll be a difficult slog to actually get this done. So here are the source data for clopidogrel. In 2000, clopidogrel marketed in 1998 in the United States, known to be a prodrug. The pathway was not known. So a clever French investigator named Jean-Sébastien Hulot discovered that it was CYP2C19, and he did this experiment in normal volunteers. He took a group of people who were wild type and a group of people who were heterozygous for a single loss of functional allele that we call STAR2, pretty common. I'll show you the frequency data in a second. And he tested them at baseline. So the y-axis here is antiplatelet effect. And then gave them 75 milligrams of clopidogrel a day for a week. That's a standard dose. And looked at the platelet function again. So at baseline, there's no difference between the two groups. But after a week of clopidogrel, the people who have the wild type genotype, they do have an antiplatelet effect. But the important point is that there is tremendous variability. Some people don't have much of an antiplatelet effect. Some people have a lot. And in here, the average, population average, is no response. But there are people who have a response. And there are lots of people who don't have a response. So that's the population average. But if you're the individual, you still don't know whether you fall into one group or another. But other things being equal, you would guess that if you were a poor metabolizer, you would be in one of those, a less adept responder. And that translates into this kind of outcome. This is a retrospective look at a large clinical trial that compared people, this is the outcome only on clopidogrel. If you carry a variant, you have a 12% incidence of heart attack, stroke, death, or stent thrombosis in the year after you get your stent. And if you don't carry a variant, it's 8%. So it's not like the difference is 0 versus 100%. The difference is 12 versus 8%. A lot of cardiologists think that that's a pretty small difference, and it's not worth the testing. And we'll come back to that in a second. Dean Eidelman said that we're doing this in PREDICT, and this is what the electronic record looks like. This is a patient of mine. The reason I know that is because I've blacked out all the identifying information, so I left this so people would at least know that I see patients every so often. And what we have is a patient who has genetic information embedded here along with their medications and all that. So that's the genetic information. And doctors at Vanderbilt are now getting used to seeing that. And at the beginning, they were not very happy to see it, and now they're more happy to see it. And what's interesting is that when clopidogrel is prescribed to a patient who has variant genetics, the prescribing system will pop up a message that looks like this, that actually it doesn't look like this anymore because we're in the next generation, but says genetic testing has been performed. Be careful with this patient and this drug. This is what the CYP2C19 genotypes look like in 13,500 patients. So about 2.7% of people are homozygotes. That means they have no active enzyme, and they really should get something else. About 20% of people are heterozygotes. You can overcome the clopidogrel resistance by giving a higher dose. And people do that, and they use the genetic information this way. And the vast majority of people have nothing, have no variant that we can find. And we went back and then looked at the 5 or 6 drug gene pairs that we were looking at and asked the question, how many people have a bad variant? How many people have a sort of bad variant? And how many people have nothing? The first column is clopidogrel. That's the 2.5 and 26% number. And the others are the drug gene pairs that you see at the bottom. And what's interesting is that when you take 5 drug gene pairs and just a handful of genes and put them together, 5% of people have something you'd really like to know about if they were prescribed that drug. And another, almost 100%, 91% have something that you would like to know about. So what I'm fond of saying when I show these data is, we're all genetic cesspools. It's just you don't know exactly what you have. And so this is the argument that says preemptively embed these data into the electronic record against the day that they need to be used. It's a big undertaking, but we think that it'll bend the outcome curve. We have a little bit of outcomes data. They're not in this slide deck. And somebody can ask me about that afterwards. So the title of the talk is Little Data and Big Data to Personalized Medicine. Let me come back to the individual patient for a second. I show this because it's an awesome graphic. And the graphic is of a big project that we have at Vanderbilt, which we call Improving Prediction of Drug Action. There are 3 projects. You can see one of them is a QT project. That's this one. One is an HLA project with Abacavir and Stevens Johnson, and another one is an informatics-based project. And I show this because we have an artist as part of the network. We don't fund him through my grant, thank you very much. But the artist is funded, and his job is to make beautiful graphics for everybody in the network. So this is what he did for us. And so the problem that I'm studying is patients with drug-induced arrhythmias. This is a problem that I've studied virtually all of my scientific career, on and off, mostly on. So drug-induced QT prolongation and arrhythmia risk is one of the commonest causes of drug withdrawal and drug relabeling in the United States to this day. So every drug that comes along, people want to know, is it likely to prolong the QT interval? Is it likely to cause torset de pointe? I can say that here properly. When you go to the States, they don't know what you're talking about when you say that. And so many drugs fail because they look like they're going to cause this adverse drug event. So it's an important thing for the regulators. It's an important thing for the pharmaceutical industry. It happens to be an important thing for the patient if they get this. You may not be a cardiologist, but this is not a good thing. And I always wonder when I show this electrocardiogram, what was going through the mind of the ECG tech as this was being recorded? Because the patient had to be unconscious. And I don't know what they were thinking, but I'm glad I have it. So the question we want to ask is, to what extent is the risk predictable in an individual patient? And the best way to do that is to get heart cells from affected patients and not affected patients and compare them. And until, when I go to people and ask them for a piece of their heart, they say no. When the oncologist goes to the patient and says, I want a piece of your tumor, the answer is no, I want you to take the whole thing. So we've not been in a position to study what the oncologists have been able to study until very recently. So there's this new technology called induced pluripotent stem cells. We can take, used to be a skin biopsy, now we just take peripheral blood, and we take mononuclear cells from peripheral blood and create induced pluripotent stem cells. Those are stem cells that belong to that patient, have that patient's DNA. They're pluripotent. We actually demonstrate that they can make ectoderm, mesoderm, and endoderm. We check the karyotypes, because the karyotypes turn out to sometimes not be perfect. And then we can drive them into the cardiomyocyte lineage. People have driven them into hepatocytes, neurological cells. We drive them into the cardiomyocyte lineage. And then the plan, and this is an ongoing project, the plan is to compare electrophysiology of these cells at baseline and after challenge with specific blockers of specific ion channels, and also look at transcriptional profiles. And we think we'll be able to do that, and then come up with predictors of which patients are at risk, and can we identify that, why. and then deploy those predictors back into the populations. So that's the plan, and we're about 1.5 years into a 5-year project. I've talked a lot about the health records bit of this, and I've talked a lot about the genomics bit of this. I haven't said a lot about a lot of other tools, but I just want to make sure that nobody walks away thinking I'm a geneticist. I dabble, or more than dabble, in genome science. But I think that if you're going to start to look at individualized health, you have to look at other measures of what the genome can do. So that's transcriptional profiling, metabolic profiling. People are very interested in engaging patients to find out what they're doing. And smartphone technologies look like they're going to be an interesting addition to that. On other versions of this slide, those are induced pluripotent stem cells in the corner, and they're beating, but they're not beating here. I wonder if they have an arrhythmia or something, but they really do beat. On my computer when I run it, they beat. And then there are other aspects of healthcare or health that we really are starting to pay much more attention to. And that's sort of things that are broadly termed the sociocultural determinants of health. So that includes, how far away from a grocery store do you live? Can you afford to come back to clinic to have your drug levels checked? Can you afford to buy your drugs? Those are all really, really important issues. In fact, people say that the most important code that determines your health is not your genetic code, but your zip code. And I actually believe that. So the challenge for us in the personalized medicine space is integrating health records and genomics and everything else to come up with a portrait of what patients really look like and how to personalize care. So I have a slide that I always end this talk with. And I have to say that I always end the talk with this slide, even though it's not this audience. But this is my slide that I always end with this quote. So I'm not trying to kowtow to Osler or the Osler-McGill roots, but the good physician treats the disease, the great physician treats the patient as the disease. This is a picture of one of our cardiologists. He has just put a stent into this woman at the beginning of our PREDICT program. And at the beginning of the program, we're of course running around like chickens without our head on, looking for the first star 2 patient we could lay our hands on, and this is her. So Dr. McPherson is taking care of this patient, and he knows everything there is to know about her desires for long-term and short-term care, her other diseases, her other medications, and now he knows a little bit more about how to take care of her. So that's personalized medicine. This is a team sport. So my rule about this particular slide is you have to be at Vanderbilt to have your picture on this slide. So there are lots and lots of people who have been at Vanderbilt and left, and sadly I take their picture off the slide. I'll only highlight 2 individuals. One is Josh Denny, who is as I said, my scientific current partner in crime for many things. And my other partner in crime is Nancy Cox, who is the developer of the Predict scan. She's the zebrafish person. And then I could say a million things about just about everybody in this picture. This is the Abacavir couple, for example. And these are all fellows who actually do all the work. And wait, gosh, where is he? There. And this is Quinn Wells. Okay, just so for you to know. So that's I told you, just grow a goatee, you'll look just like him. And this is institutional leadership, without whom none of this would ever happen. So usually I stop there, but I spent a lot of my time at this university, and I was telling the dean on my way over here, I actually lived in the student union. He said, "Oh yeah, I didn't even know there were apartments there." There's 7 apartments. One of them belongs to the managing editor of the McGill Daily, which is how I spent the 1968-69 academic year, living in that apartment. I also ran for student council that year. This is my student council running picture. Taken by a very talented photographer on the McGill Daily staff named Nick Dykeman. Nick took that picture. I lost the election. But what was interesting, I was actually on the web literally a couple of days ago, just sort of looking for things to say at this point. And what was interesting, and I needed to be reminded of, is that there were a bunch of people who ran in that election, most of whom won, and there are names that you know. So George Roglansky was Privacy Commissioner for the country of Canada. Charlie Krauthammer, a very distinguished political commentator from the other end of the political spectrum, but very distinguished nevertheless. This was before his accident where he broke his neck. And Norman Spector, who for a while was Brian Mulroney's chief of staff and ambassador to Israel. So those were all, and in fact, if you look closely at the Norman Spector thing, it says, paid for by the Spector-Roden Committee to elect Norman Spector. Anyway, so that was how I spent 1966 to 1970 here, and then I went to medical school and fell in love with medicine. Although I didn't fall in love with what I do immediately, but I was really, I'm really very grateful to McGill Medicine because it sort of made me who I am. And I'll close with this picture, which I'm fond of saying. So this is my home away from home for many, many years. Our oldest son, my wife is here, and our oldest son was born at the Women's Pavilion at the Vic, and I have extremely fond memories of this place because it really did set me on the right path. So thank you so much for asking me to give this talk.
Congratulations. Thank you very much for that extremely clear and extremely enlightening talk. I want, before entertaining questions, I want to just thank a couple other people. I want to thank someone named Ray Hakeem who nominated you for the Holmes lecture. I don't know if he's here right now. I don't see him. But he'll be around, I think, this week. And I also wanted to thank Dimitra Kafantaris and Sarah Smith, who are here, who did all the work.
They've done all.
And with that, I gave people a chance to think and cogitate and digest, and I see if there are any questions.
No questions. There are never any questions.
So I have a question while we're waiting for them to think. So I thought this was a brilliant plan to create this enormous database. The question, when I was walking over here, we were talking a little bit about ethics. And in this province, for example, we have very, very strict privacy laws. And when you obtain samples of any kind, in fact, initially when the law was first passed, they claimed that microbiota were actually the property of the people from whom they were swabbed, but they changed that. Okay. So we actually are allowed to culture microbiological material independently of other species. The question is, how do you deal with the ethical issue of discovering something or doing experiments on samples of some sort, whether they're of any kind, after the fact? How do you construct a consent which is accepted by the authorities for that kind of experiment?
So I glossed over a lot of things, and that's one of the things that I was happy to gloss over. So I come back to this, and I just want to highlight this person here, Ellen Wright Clayton, who's a distinguished, nationally recognized ethicist. And my wife and I just moved, but she used to be our next door neighbor. Now she lives about a mile away. And so Ellen has been a driver for us and in the national scene to understand how to do this. Our biobank is unusual, and I don't know whether it's a good thing or not, but all the DNA is coupled to de-identified electronic records. And the rules of engagement right now, and they're not going to change anytime soon, although we're thinking about how to change them in a new genomic world, are that once you're in, we make zero attempt to figure out who you are. And in fact, it is one of the major pieces in our data use agreement is that you're not allowed to try to figure out who somebody is. And if by accident you actually see a record that's de-identified, but you can still figure out who they are, you're supposed to tell us. We call that the astronaut's mother syndrome. So, the history of President Elman starts with this 79-year-old mother of an astronaut, blah, 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 blah. So
that narrows it down.
That narrows it down, right. Or better yet, this 45-year-old ex-astronaut. So there's some things that you can't sort of get out of the record, but those are things that are there. So, this 89-year-old vice president's mother.
Yeah, that's a small sample.
So those are, but basically the data are decoupled from the patients. So we're fond of saying we don't have 237,000 patients, we have 237,000 avatars, and they sort of represent what a patient might look like. That said, we are increasingly, as we start to sequence people, as we start to do sequencing as opposed to just genotyping, we will find people with BRCA1 variants. We will find people with ion channel variants. And I used to worry a lot about, I still worry a lot about that, but I used to worry more because what's clear is that all of those things are highly impenetrant. So if you have a strong family history of breast cancer, you don't need us to be dealing with you. You need to go and tell your doctor. The problem is that there are many people who are, de novos or who don't have big families and who then discover, and there's a big controversy in the whole genomic medicine world about whether people should be getting BRCA1 testing, for example, as a routine to find, patients who are carriers. And that controversy brews because even if you have something, it's not clear that you're going to get the disease. Sure. So the answer, I'm skipping around the answer. The answer is for the biobank, we actually are sort of off the hook, but at the end of the day, we're probably not completely off the hook. But when we start to do studies where we're actually sequencing actual patients, there's a whole consent mechanism around that. And then the difficulty becomes, if you're a cancer patient and you're screened for cancer susceptibility genes and your tumor is sequenced and somebody discovers you have an ion channel variant, what do you do? Mm-hmm. And there's so the American College of Medical Genetics has a list of 59 of their least favorite genes, and they say if you have a known pathogenic variant in this gene, you should return it. But the known pathogenic part is the operative part, and it's very, very difficult. Of all the hundreds and hundreds of variants in every gene that you'd ever look at, the ones that are known pathogenic are a really tiny list. So it's a huge issue, not just for us, but for anybody who's starting to do population genomics. The incidental finding, and then the variant of unknown significance, and they're completely related to each other.
Okay, Demetrius pointing in that general direction. Is there a question? Oh, there it is. It's Al. Okay, sorry.
No, and also.
Yep. We'll start. I'm recognizing Dr. Schreier first.
So, on this topic, when you take the iPSCs from a patient, you screen them, look at the response to different types of drugs.
Wait, just hold off because we need you to speak into a microphone for YouTube.
For YouTube. Yeah. You sure you want to say? You sure want to ask? I don't know.
Anyways, great talk, Dan. So if you get the iPSCs from patients and now you're going to take them and challenge them with drugs and look at the change in their electrophysiological properties, you're not considering going back and saying, this drug will work on that patient or that drug won't work. So do you not take that information and go back to the clinic with that?
Oh, eventually we will. I mean, but this is developing those kinds of predictors. I talked to somebody today, and if you'd asked me 10 years ago, I said, I would love to find the SNP that predicts that response, and that's sort of a pretty naive view of the world. I'd love to find the set of markers that include SNPs, include metabolomics, include whatever, the signature. Thank you very much. And it could be just a genetic signature, because maybe lots of SNPs though. And come back and say that. And what we'll do is we'll take that information and deploy it in the electronic record. All these IPS patients are consented, of course, consented out the wazoo. And we'll do whole genome sequencing on them to find if they have anything in the long QT domain or anywhere else. And we'll do the transcriptional profiling, which of course, that's a totally research effort because no one knows, exactly what it is you're sort of expecting in that kind of space. So yeah, we will go back. But it's one of the questions that I get asked that's much more pedestrian than that is somebody has an episode of torsade de pointe, a long QT related polymorphic tachycardia on a drug. Should they get genetic testing for the congenital long QT syndrome? And we think the answer is no, unless after all the dust settles, their QT interval is long. If their QT interval is totally normal, we have data that says that they probably don't have it.
Okay, thank you very much. Another question right here.
I'll try to be shorter.
Dr. Diachenko.
So are all these data sets available in public domain, or they're only available for the Vanderbilt researchers?
Yeah, so the data sets that we generate as part of our work with eMERGE and other consortia, those are all by law and by design deposited in dbGaP. And if you want to get to them, you can get to them through dbGaP. It's a pretty cumbersome process, but there it is. The problem with us making the entire data set available publicly is that people do look at the electronic medical records that are de-identified, and people sign a data use agreement preventing them from trying to do re-identification. And if they do, there are sanctions. People are allowed to be, have their access denied. People are allowed to be fired. But if an external user came along with, we don't have any possibility of sanctioning. So we don't know what to do with that. It's an interesting problem because the Precision Medicine Initiative is going to do this on a huge, huge scale with identified samples. And they'll have a data use agreement, but if you have a user who uses the data in a way that they're not supposed to, it's hard to conceive of what kinds of sanctions the government or anybody else can impose.
One can certainly imagine insurance companies and private detectives and any number of other not so friendly people.
Oh, no, so we had a lecture. So I'm as you imagine, I participate in a lot of activities around this at our place, and we have a set of I'll say Ellen and Brad. Brad is the privacy ace. They have a grant together to think about privacy in this space, and they had a big meeting of that grant last week, and we had an hour-long lecture on what the law allows police authorities to do when they come to our data set. What can they ask for, what can they not ask for? So it gets very, very complicated very quickly, and there are times when I thank my lucky stars that we're a de-identified resource. So we state publicly that we cannot go back to who the patient is, and that's the position we have. So as long as we keep that position, then nobody can come in and sort of say, I want that person's data, because we don't know who that person is.
Dr. Thanasoulis.
Dr. Roden, that was a fantastic talk. Thank you very much for that. I guess a question I have is, I think in Canada we're probably a little bit behind on genomic medicine. McGill, for example, has a long history of genetic research, yet we don't actually have anything like the BioView biobank. So my question is to you, do you think that at this time, given that the Precision Medicine Initiative is starting in the US, is it something that we should even consider doing? And if we were to do it, how would we start and how much would it cost?
You and I are going to have dinner tonight, right? So we can continue. There's no short answer to that. BioView is pretty cheap on a grand scale. BioView, I'm the PI, so I know what the budget is, and it's somewhere. At the beginning, it was about $500,000 a year, and now it's about $1 million, $1.5 million a year. And the number of grants it's brought in is, if we count the Precision Medicine Initiative, it's somewhere north of $150 million. So it's been a worthwhile investment. So, and I understand that not only are the dollars different, but the calculus around those dollars is very different here. I don't think it's too late to get into this. I would say that one of the models that I find appealing is there are collections of people at your university, because there are collections of people at every university who have 1,000 samples of this and 1,000 samples of that, and so finding some way to federate those as a beginning. So you have 10,000 or 20,000 people, and then you have to have informatics chops to sort of figure out how to bring them together and harmonize some of the stuff. But at least that way you have a sort of a starting set. That's a model that appeals to me. And then this, ours is based on the fact that we have this electronic medical record. I was telling the dean on our way over here that we had a vice chancellor who said in 1991, we are going to be informatics central, and we invested in a medical record early. It's our own medical record. We're not beholden to anybody else. And I wish I could keep on saying that, but for cost and other reasons, we are switching to Epic. And so when I try to talk to my biomedical informatics friends to get them to do something for me, the answer is always, I can't, working on Epic. And after November 2nd, maybe they'll be able to. Something you said earlier to me reminded me to say this, that the trouble with Epic is that if you've seen one Epic installation at one hospital, you've seen one Epic installation at one hospital. Yeah, so they're all different.
It's a great business, though, Epic.
Oh, geez, don't even start me. But an electronic health record that captures outpatient, inpatient, all that stuff. I think that the Precision Medicine Initiative that we're doing in the States, I think, is predicated on the fact that eMERGE, this network that we're part of, showed that the electronic record could be a source of dense phenotypic information. And so that's been the propeller, because it feels like it's going to be cheaper, and it feels like it's going to be directly health related, because people go to the hospital for some reason as opposed to some questionnaire. But we'll do both in the Precision Medicine Initiative. So I can't tell you if it's too late or too early. I will say that that Canada has made very interesting investments in genome science, and some of the projects that they've invested in are things that we look at and we say, gee, we wish we had that kind of chops as well. So that's a two-way, the jealousy is a two-way street.
So Ariane, actually, Dr. Burke had his hand up first, so, no, it's actually, Guillaume gets first dibs, then I had Dr. Fuchs, and then Dr. Morelli.
It's sort of a follow-up question. So you showed the results of setting up the infrastructure to do especially the genotype, phenotype associations and the great results there. And then you touched a bit on all the other types of data sets that are out there. And so I was wondering in terms of what you think are going to be successful story out of those, and what are the most promising data sets, whether it's imaging combined with phenotype as well?
Well, so we're constrained in BioView because of this de-identified business. So we have thought a lot about getting samples for metabolomic analysis, for example. The problem is that we use convenience samples. We capture the samples when somebody's had a blood test done. The sample sits in pathology for 3 days, and then we grab it as it's going to the garbage can. So that's not suitable for metabolomics for two reasons. One is that the metabolites that you're interested in may not have survived that 3 days. The other is that you have to ask yourself, so why did that person get that blood sample in the first place? Do they have a temperature of 105? Do they have a broken leg? Do they have something going on? Or is this just a well baby check for that person? And we obviously like the second and not the first. So the plasma stuff is a little harder. The imaging stuff is very, very interesting. Relating imaging to genomics, and we have a huge collection of MRIs that are all digital that lend themselves to being de-identified. The problem is the storage problem that we talked about earlier today. It's just, so you think a genome costs a lot to store, just wait until you try to store 1 million MRIs.
So we do have, although we don't have this kind of project, we do have some project that we have C-Brain here, which is sort of that kind of project for brain scans, and it is a huge data storage problem.
And just imagine trying to then couple the EMR, the images, and the genomics. I would say it's a nightmare, but it's a sort of something we'd love to do if we could figure out how to do it. And then all the sociocultural stuff, I think we're going to have to do by questionnaires. And the metabolites and the proteomics, I think you have to do that in a sort of who am I doing it for and why.
Hey.
Thank you for a really beautiful overview. I mean, clearly your group is very careful in what they say and do, but in the marketplace,
Those guys.
not only 23andMe, but I heard just the other day a company called Telomark or something, where you can measure telomeres, and it's almost like a custom tailor service, except they don't tell you what the dress will look like. So
Right. Do they try to build your telomeres back?
That's right. And then give you advice. So is it the case that these companies, despite the care with which the scientists approach these problems, are going to create an epidemic of what you might call incidentalomas, not now in imaging, but genetic incidentalomas or telomeric incidentalomas? And how are we going to catch up to commerce that is really unregulated?
So I'll make a correction. The collection of incidental findings is actually termed the incidentalome. And I didn't make that word up, but I have friends who made that word up. The word was made up by Russ and Zach and Dan Masis in an article in JAMA. So my wife knows those people, and that's fine. So they actually wrote about the dangers of the incidentalome about 10 years ago. The 23andMe story I find really disturbing, and here's an anecdote that says why. I was actually asked to do a radio talk show at 5 in the morning Central Time. So I'm up at 5 in the morning and I'm taking these calls, and this woman calls. It was right at the time that the federal government had said to 23andMe, no more advice. This was 4 or 5 years ago. And she said she was really upset because she had a sister with breast cancer and mother had died of ovarian cancer, and there was an aunt with breast cancer, and she really wanted to get the results of her test. And I said, well, I think I got it right even at 5 in the morning. I said, first, you need to be seen in a cancer clinic which specializes in this. And the other thing is that the 23andMe test for breast cancer tests 3 common variants in BRCA1 by genotyping. It doesn't sequence BRCA1. It doesn't sequence BRCA2. It doesn't know what to do with the variants of uncertain significance. And there's a whole bunch of other cancer susceptibility genes that they don't look at. So when people say, my concern is when people say, I was tested for breast cancer and I don't have any risk alleles, that's a false negative.
Yeah.
That's the
Especially if
thing
they have a
I
family
really,
history.
really worry about. I mean, I can't, I think that's the much more dangerous thing. So and I agree with everything else. And maybe we should have laws regulating this in the United States. That's a little joke. Yeah, right.
No comment on that one, but we do want to hear the last question from Dr. Morello.
Right.
Thank you so much for such an enlightening talk. I have a comment and actually, to finish this off, a question and a challenge for our dean. We have unique data sources in Canada, particularly in Quebec.
Yep.
For example, in congenital heart disease, we have an integral population-based
Right.
data set, which follows patients for 28 years retrospectively, because we have unique identifiers from birth. We've been able to get this administrative data from the government, and it's different from NIS data because in our cases, they're actually patients, and we have 28 years of electronic information on them. And this is singular, and I work with the CDC, and they're looking at this and going, how do we mandate, how do we change policy for patients with lifelong conditions so that they can get unique identifiers at birth in the United States, so that this kind of data can be generated. So for me, this is actually an incredibly interesting opportunity. The question I have for our dean is, we need to start lobbying our policymakers to allow us to do things like this. It would be so incredibly, I'm not going to say easy, but it would be so incredibly rich for us to be able to say to RAMQ and Medico, for example, I'm just getting the last 5 years of data until 2015. I need some kind of permission to collect blood in a de-identified way. We would have such a wealth of data.
So,
And we need to think from, we need to think, I think, as a university, with our faculty of law, with our faculty, with our ethicists, we need to put a group together that's really going to go to government and say, we are at a crossroads, and we can do this, and we can do this now.
so Ariane, I think you're absolutely right. I have good news, however, because although we haven't done exactly your project that you've talked about, Philippe Gros, who unfortunately can't be here, has been working with colleagues at the University of Sherbrooke on a birth cohort that they are actually looking at in sort of this way. And there has been openness with support from the Genome Quebec and also the FRQS and the chief scientist, Rémi Quirion. So I actually think there is more opening for that now. With the advent of the so-called SPORE, the SRAP, there is an interest in having more money available for so-called patient-centered research. And this is a good example, actually, of patient-centered research. And I think that there is much more opportunity in defined cohorts like congenital heart disease than there is in the general population because of it's much easier to make the ethical argument because these are people with very specific needs. And so the ethical balance, the equipoise, is in a different position than if you just take random people like 23andMe does and says arbitrary things. If you have a group of people who are identified, cystic fibrosis patients, patients with childhood rheumatological disorders, you can just imagine a set of many different groups of people who are going to have well-defined risks, but who need more attention. And there have been people at McGill who study things like this, like the risk of malignancy in people with rheumatologic disease over the long term, and things like that. So I actually think you're right. I'm not sure I know everything's my job, but I'm not sure that this is actually my personal job. But I'm ready to help. I'm certainly ready to help champion this because you're absolutely right. We do have, I just want to say that although we don't exactly have what they have at Vanderbilt, we do have a number of things at this university that are quite cool and are parts that are complementary in many ways to what Dr. Roden has presented, and we should also take advantage of that. And so, our neuroscience group has a very powerful imaging sort of approach. Our genome center is a world-class genome center, to put it mildly. And I do encourage, in the same way that you're encouraging the dean to champion this, I encourage our faculty members to think a little bit more broadly about collaborations that we can do even within our own institution. On that note, I'm going to officially thank you, and I have a small presentation that Sarah is going to get for us. And we'll actually do the picture taking afterwards. But we give. This is a plaque. It basically says that you have received the Andrew F. Holmes Dean of Medicine Distinction Lecture in April 2017. So it's my pleasure to offer you this as a token of our appreciation.
Thank you so
We'll
much.
put it back in the box so you can transport it home.
Yeah,
With that, this brings us to the end of our official activities. I want to thank everybody for coming out, particularly in the middle of one of the least pleasant days we've had in a while. Have a great evening. Thank you very much.
Thank you very much. Thank you.
So we'll put that back in the box.